Hello students, myself Om Prakash Katte and you are welcome to Biology Reward. Today we are discussing class 12th chapter Molecular Basis of Inheritance, and today our topic is translation. Translation is our data. Earlier we have discussed regarding the transcription and genetic code. Now we have to understand the process of protein synthesis. It is translation. Students, we have learned about the central dogma, and this central dogma we have learned molecule of DNA, it transcript to form the RNA, and then the RNA molecule is translated to the protein. So this phenomenon is called the central dogma. So this RNA molecule, which is having the genetic code, this genetic code, they are synthesizing the amino acid. Genetic codes are present in RNA molecule. This RNA synthesizes the amino acid or amino acid. This amino acid, they form the long chain which is called polypeptide chain. The chain is called polypeptide chain, and this polypeptide chain, it converts into the protein. And simply the RNA, it get translated to the protein. This phenomenon we are discussing, and this is the translation. So in protein synthesis we have two processes. One protein synthesis, two process we have. One transcription. Transcription, what we have learned? We have learned the DNA molecule, it transcript RNA. So one strand of the DNA, it get transcript to the RNA molecule, and we have studied about transcription in prokaryotic also and eukaryotic also. So this is the first part, transcription in protein synthesis. Second part is now this RNA, it code the protein. It means RNA, it get translated into the protein. So this phenomenon is called the translation. And thus protein synthesis, we have two phenomena, one transcription and translation. So next we are discussing now translation. Translation is what? The definition of translation is the process of polymerization of amino acid to form polypeptide chain, and then it forms the protein, and this is called the translation. So what is mean? Amino acids, RNA is having the genetic code, and this genetic code synthesizes the amino acid. Genetic codon, it forms the each codon from the one amino acid. So this amino acid and this amino acid, they connect to each other, and that form the long chain. Suppose this amino acid, and if this amino acid are connected, so the long chain is formed. It is called polypeptide chain, and this polypeptide chain is converted into protein. So this phenomenon is called the translation. Process of polymerization of amino acid. It means many amino acids are combined and they form polypeptide chain. Then this polypeptide chain is converted into protein. So this process is called the translation. Okay. So many amino acids, they combine and they form the long chain of polypeptide chain, and then this polypeptide chain convert into the protein. This phenomenon is called the translation. So we have to study the process of translation step by step. Translation process is little bit complicated process. So we have to study it step by step. Step of translation. In step of translation, first is activation of amino acid. Students, you should get the one idea. First you see the cell. This is the cell, and in this cell, inside the cell there is a nucleus. This one part is the, so cell, inside the cell there is a nucleus. Inside the nucleus there is a chromosome. Inside the nucleus is a chromosome. Then chromosome is made up of DNA. So chromosome is made up of DNA plus protein. You will see that in nucleosome, madam, we have learned the DNA molecule is wrapped by the some octamer unit of protein. So chromosome contains the DNA. This DNA molecule is converts or it transcribed to form the RNA. So cell, inside the cell is nucleus. In nucleus there is a chromosome. In chromosome in nucleus is DNA. So this part is in the nucleus. But when DNA transcribed to RNA, this RNA are formed and this RNA come in the cytoplasm. So this RNA it's come in the cytoplasm of the cell. From nucleus, this RNA come in the cytoplasm. So what happened? In the cytoplasm only, there are large number of amino acids are occur. So large number of amino acids are occur in the cytoplasm. So what happened? For translation process, first one, in cytoplasm, there are amino acid are there. This amino acid are in the inactive form. So this in cytoplasm there are amino acid. These are the inactive form. So first one had happened. The amino acid which are present in the cytoplasm get activated by using the energy in the presence of amino acid synthetic enzyme. Amino acid, they are using the ATP energy, and in the presence of amino acid synthesis enzyme, it's from the one amino acid AMP enzyme complex. So this substance is formed. This complex molecule is formed. Amino acid activated by using the energy in the presence of amino acid synthesis. So it is called activated amino acid is formed. Amino acid plus AMP plus enzyme complex. So in the presence of amino acid synthesis. So this is the first one. In this phase, pyrophosphate is released. What is the stage? What is the activation of amino acid? It is the process in which amino acids get activated by using the energy in the presence of amino acid synthesis, and what is formed? It is amino acid AMP enzyme complex is formed and pyrophosphate is released. Now this is the amino acid synthesis enzyme. This amino acid synthesis enzyme, here I am making the one diagram. This is the enzyme, amino acid synthesis enzyme. So this amino acid synthesis enzyme, here what happened? This amino acid group is combined here. This is amino acid is combined here in the amino acid synthesis enzyme, and here amino acid is combined. So the amino acid first get activated, and this activated amino acid is combined with amino acid synthesis in one side. Now second, this amino acid AMP enzyme complex, it means this amino acid and enzyme complex is combined with tRNA. So here is the another side. Here is another side where the tRNA molecule is combined. Here is amino acid and this here. So total what we are getting? This tRNA molecule and here amino acid is combined. So in this process, amino acid synthesis, what is the function of this one? First one, amino acid synthesis activate the amino acid and it helps to join with the tRNA. So transfer of amino acid to tRNA. This is the second. This amino acid and enzyme complex is combining with the tRNA. And this one is from the amino acid tRNA. It means amino acid is combining with the, so if you see the molecule of tRNA, this is the structure, and in this molecule of tRNA, here is a side, CCA side, this is the third part. So in this side, the amino acid is combined. So it is called amino acid is attached with the tRNA with the help of amino acid synthesis. So first process, amino acid get activated in the presence of ATP and amino acid synthesis. Then this complex is from AAMP enzyme. This charge now with the tRNA, or it combined with the tRNA, and then amino acid is attached with the tRNA molecule. So there are large number of tRNA molecule which bring the different amino acid. So large number of tRNA molecule, and they bind, they attach with the amino acid, or amino acid are attached with the tRNA, and many amino acid are brought by the different tRNA molecule. So this is the process number 1. Here we are getting amino acid tRNA. It means amino acid is connected with the, attached with the tRNA molecule. Second process is DNA is changed to mRNA. This is the messenger RNA. So in messenger RNA, here there is a one codon AUG, which is called initial codon, which code for the methionine. So first we are talking small ribosomal subunit 30S attached with the mRNA. This is the mRNA molecule. So in mRNA molecule, ribosome, prokaryotic ribosome, you should understand. Prokaryotic cell, they have 70S ribosome. And this 70S ribosome is divided into sub-unit. One small unit is 30S and another is 50. So it is prokaryotic cell, 70S ribosome. They have small unit is 30S and another unit is 50S. So what happened after this? First one, small ribosomal subunit 30S attached with the mRNA molecule. So look at here, this is small ribosomal unit. I am making the one diagram. This ribosomes, small unit, and this is the big unit. So these two units get separated. They are dissociated before the protein synthesis. So first small unit, 30S unit of ribosome, it attached with the mRNA. So small ribosomal subunit attached with the mRNA, and this is now from the 30S mRNA. Now second, after this, big ribosomal subunit, which 50S unit, this 58 unit attached with the, so it is the 50S unit and this is the 30S unit. So here 30S plus mRNA plus 50S unit, then 70S mRNA is formed here. So first one stage, activation of amino acid, that activated amino acid combined with the RNA. Now the when small unit combined with mRNA, it is from 30S mRNA unit, and now the 30S mRNA unit, then large unit is combined, it is from the 70S mRNA molecule. So here this is the mRNA molecule. First is 30S unit is combined and then 50S unit is combined. Now one more thing we have to understand, and this is regarding the amino acid synthesis. As I told you, the amino acid synthesis is this one molecule. In this site is the one corner, one point is here. Here activated amino acid, it means amino acid plus ATP. This activated amino acid are combined here. Then tRNA molecule, this tRNA molecule also, this is the tRNA molecule, and this tRNA molecule is combined there. So when the activated amino acid and tRNA, now the new combination is formed, the amino acid tRNA. So this is ready in the first one steps, this activation and transfer. And second step is small unit combined with mRNA, and then 50S unit combined with 70. It means second, this one, small unit is 30S, and then it is the big one unit, it is the 50S. So this is the first important step, and this is the second important step for the translation. Now we have to go for the process, how this translation process is occurred. So in this translation process, first point is here, initiation of polypeptide chain. So during the initiation of polypeptide chain, what happened? 30S subunit bind with fifth end of the mRNA. So this is the mRNA molecule, and this is the fifth end or fifth prime, and this is the third end, it is the third prime. So this in this process, one physician, when initiation is start. So as I told you, this is the one enzyme which is called amino acid synthesis enzyme, and here activated amino acid and there's tRNA. So the amino acid with tRNA. Okay, so this is in enzyme. This enzyme move from prime fifth to prime third. This acid, it move from prime 3 to prime 3. Fifth end to third end, or prime fifth to prime third. So in this direction, the movement of this enzyme take place. Now, initiation of polypeptide chain. For initiation, this is the fifth prime. In this fifth prime, there is a one codon. It is called AUG. It's an initial codon. So in this time, in this codon, here is a small subunit of ribosome. It is 30S unit is combined. So 30S subunit bind with fifth end of the mRNA, where AUG code, which code for the methionine is here. So 30S mRNA is formed. Now, this tRNA molecule, which is called the amino acid, and this molecule is the tRNA molecule. So the tRNA molecule, this first tRNA molecule is combining with the methionine. Methionine, because methionine is the AUG codon, the AUG code for the methionine, and this methionine is function with the formyl group. So it is called formulated methionine. Understand first. First one codon is AUG, and this AUG code for methionine amino acid. So this methionine amino acid get functionalized with the formyl group. So when methionine get functionalized with the formyl group, so it is called formulated methionine. So this tRNA with having the formulated methionine, so this tRNA is combined, amino acid is methionine, so methionine containing tRNA, it's combined with this one side. And here anticodon will be, A will combine with U will combine with A, and G will combine. So here anticodon is UAC. So first one, small ribosomal unit, 50S subunit bind with the fifth prime or fifth end of the mRNA, where AUG codon is there, and this the fit code for the methionine. Second, this is combined here. This tRNA with methionine combined. After this, the large unit, which is called the 50S unit. This large unit is combined. Now what to understand? We have to understand the large unit having the three sides. The large unit is having this is the small unit, large unit having this is called E, P and A side. There are three sides. So now when fifth S unit, it means large unit, has E exit site. This is E is for exit site. P is for peptidyl site. Peptidyl site. A is for acceptor site. So, what are the function of these three? You must understand. A site is called the acceptor site. It means whatever amino acid will come, whatever amino acid will brought by the RNA and tRNA. So, this tRNA will bring the amino acid step by step. Different types of tRNA will bring the amino acid, and this amino acid combined with the A site, and that it is called the acceptor site. Second, P is called the peptidyl site. Peptidyl site means in this site, this methionine tRNA is combined. This tRNA, first tRNA is combined here. So it is called methionine tRNA site. In this site, peptide bond formation takes place. This is the site where amino acid will attach. In this site, methionine is attached, and here peptide bond formation takes place. So this is called peptidyl site. And E, if the peptide bond is formed, then this tRNA is exit out from this one side. So this side is called the exit. So we should understand here, there are three sides. First one is the E exit side, from which the tRNA molecule will be removed. One is the P side, where the peptide bond formation takes place, and there is A side where the amino acids are attached. Now, 50S subunit bind with the fifth prime. Now this one, see, use simple diagram. This the molecules of mRNA, fifth and third end, and here the amino, this small one unit, 30S is joined. Then this is the 30S unit, this is the one A site, this is P site, and this is the existing site. Now, 50S subunit has three sites, E, P, A site, exit site from which the tRNA molecule after forming the bond will be released. P is peptide, where the peptide bond formation takes place. A is acceptor, where amino acids are combined. Now, this bind with the 30S unit, it means 50S and 30S, these units are bind. Terminated methionine tRNA bind with P site, and other amino acid bind with the A site. So, only one amino acid here, this one says, which I told you, in this P site, only tRNA with amino acid methionine. So, methionine tRNA only bind with the P site, and other amino acid they bind with the A site. So, it is exception. Only one amino acid, first amino acid, it means formulated amino acid will bind with the P site, and other amino acid will be bind with the A site. So, this is the first process. What to understand here? In this process, initiation of polypeptide chain takes place. First one on mRNA molecule. First, fifth prime, it means fifth end, 30S subunit combined, where the AUG codon is there. Then, after this, the 50S, it means large unit, which is having 3 sides, E side, P side, and A side, it's combined with the small unit. And this formulated methionine, so the tRNA having the methionine, it's only bind with the P side, and remaining amino acid will combine with the A side. So this is called initiation of the peptide chain. Second process is elongation. In elongation, what happens? New peptide bond is formed between amino acid of the P side and A side. So now we should understand, this is very easy. This is the P side, and amino acid is this one, R, C, H, NH2, COOH. This is the one amino acid. And A site, other amino acid I am writing. CO, it is NH2. Then it is NH2, R, CH, COOH. So this is one amino acid and this is other amino acid. So P site is this is the DNA, mRNA molecule, this is the small unit and it is the large unit. So in large unit this is E and P and A. So in P site, this one is amino acid is here. RCH, NH2COOH here. And A site, another amino acid is here, this one, this. So now what happened? Between this one, COOH and NHH, if it is written, then what happened? This molecule of water is released, so it will come NH. Okay, so this bond formation take place. So the structure will be this like R, CH, CONH, then R, COOH, and this one. So this bond is called the peptide bond. So here during the elongation, new peptide bond is formed between the amino acid of P site. This is the amino acid of P site and this is amino acid of A site, and between this one bond is formed, peptide bond is formed. Now step by step, amino acid tRNA bind to the A site and catalyzes by peptidyl transferase and elongation factor. Now next one steps we have to go for the elongation, and this is what this mRNA molecule, this is the small unit, large unit. In P site, as I told you, this one peptide bond formation takes place between amino acid of P and A site. So in this one, two sides, this peptide bond is formed. So when peptide bond is formed, now what happens? The tRNA molecule, peptide bond is formed, it means amino acid is used. So the tRNA molecule will shift to A site. It means, so if this is the amino acid with the tRNA, it is in the site P site. Amino acid is used here and peptide bond is formed, so it is the A site. A site is another amino acid. So this P site is one amino acid and A site is other amino. In between what happened? In this one peptide bond is formed. So when peptide bond is formed, it means this amino acid is here used. So this become empty. So this tRNA molecule now empty. So it comes in the P site. So thus now what happened? This is the one amino acid. Now other amino acid also will come and this will bind with the A site. Again this will shift to the P site and tRNA will remove. Next one amino acid will also come. So this process continuously amino acid will pick up by the tRNA because there are many tRNA molecules and they pick up the specific amino acid and they bring it in the A site. This A site amino acid will combine with the P site. Here peptide bond is formed and after the bond formation tRNA will shift. It is called the empty space. Now step by step amino acid tRNA bind to the A site. It is catalyzed by the peptidyl transferase enzyme and elongation factor. After this what happens? Empty tRNA. It means the amino acid is formed peptide bond. So empty tRNA bound with the E site. This come in the E site and thus long polypeptide chain is formed. So here the long polypeptide chain, this type of long polypeptide chain will form and this long polypeptide chain is called will form the protein. So this process. So peptide chain formation takes place from the 5th prime to third prime. Or we can see these diagrams in given our book. So this mRNA, small unit and the large unit. So here you will see this type of molecules of amino acid, long chain of the polypeptide chain is formed. So it is the A side and this is the P side and this is the E side. So here from the P side only one amino acid is attached. So this amino acid which comes and combined with the A side, this and this, they both will form the peptide bond, and this long chain of peptide bond is synthesized, and this is called polypeptide chain. And then this polypeptide chain will convert into protein. And third step is termination. This process of protein synthesis will be continued. Termination by any one of the stop. If any nonsense codon comes, stop codon comes, UAG, UAA or UGA. Out of 3, any one codon comes, then this translation process is stopped. And this also, when the process is stopped, what happens? In the presence of releasing factor R1, R2 and S. So once the process of translation stop, process of polypeptide chain synthesis stop, and when it is stopped, when any one terminal codon comes there. So when the process is stopped, then what happens? These 2 ribosomal unit dissociated. Then the mRNA, small ribosomal unit which was attached with the mRNA, that will be separate. Both unit will be separated. Okay, so this happened in the process of translation. In process of translation, some stand of the RNA. This is the RNA. So most of the segment of the RNA translated into protein. But there are some such type of segment. They are called UTR, untranslated region. This UTR, it means these are the region which are not translated to polypeptide chain or protein. So this UTR are untranslated region. It means they are not synthesizes the protein, or they are not the part of the protein synthesis. So this UTR region, they help for the efficient translation. This is are responsible for the efficient translation of the protein synthesis process. So in translation, definition, activation of amino acid, activated amino acid combined with the tRNA, then 30S unit combined with mRNA, then tRNA molecule will combine with the mRNA unit, then it's covered with the 50S. So when it is done, then process initiation begin. In this process, small unit combined with the 5th prime or 5th end, and where AUG is there. Then 50S unit having the 3 side, E side, exit side from which the tRNA are removed, and P side where peptide bond formation take place. A is the site where amino acid are attached and formulated methionine tRNA. tRNA which bring the methionine, it combined with the P side, and remaining amino acid combined with the A side. So the amino acid of P and A between peptide bond is formed, and thus many amino acid, when they are coming, then large number of peptide bond are formed, and that from the polypeptide chain. So this formation of polypeptide chain takes place from 5th to 3rd end, and this process will be continued till the stop codon come. If any of the stop codon come there, the process get terminated. So translation process is stopped. So this is all about the translation process.